Cellular infectious agents. When we say a cellular, they are those microorganisms that doesn't have cell. Those are organisms that nabubuhay lahiya pag nakakasulod din cell. So, the common infectious agents we know that is virus. What is a virus? A virus, it is a package of genetic material. And virus, it is either deoxyribonucleic acid or DNA or ribonucleic acid or the RNA. And virus, it has a shell covered called capsid. Tas ang ibang nga virus, may daniya extra envelope pa nga naka-cover pa capsid. So pag ang virus is waray pa hiya nasudlan na cell, tawag ito ya extracellular pa, meaning wala pininihimo, then just hanging around. Pero pag ang virus nga ni makabilinghan host cell, nagiging intracellular na ito ya, dindida na yan nag-i-start pag kalat. So ang virus nagmumultiply hiya by replicating. Tapos it virus na sulod dito yung cell, then hiyanat na control ha cell. Tapos na-mention naton ngadto na an usa nga function ng carbohydrates is nakaka-reproduce ang cell. So amitiya an virus pagdadamon sugar itaton lawasan or pagdadamon sugar ito sa living organism, dida ito ya at cell it sugar amot na bulig at virus na magdamo because amot na kwa na cell. So bali an virus is nagtitikada mo lahiya cell. Tas an cell amot talaga amot gudan na reproduce. Even though naton lawas is made naton natural defenses against virus, pero pagdadamo once nga nadamo nat virus, dida na iton naton bisan na naton immune system is di rin niya kaya na labanan. So the common example of virus is chickenpox, cold, measles, HIV, rabies. And we should also know that virus is infectious agents made up of viroid and prions. So how are viroid and prions similar to virus? And paano hira naging different? Viroid and prions are also tiny but powerful. They can both take down plant life and the entire animals. And they are also cellular particles. Cellular means that in a cell. They don't transform energy. They don't grow or divide. And they can't reproduce on their own. Cellular particles can hang around forever. Pwede ra ada doorknob, ada igbon lamisa, or bisalahirahain, and causing no harm. Pero, like what I've said kanina, once ganin na makasulod na hirahit sa kasel, di na hira nag-start pagdamo. And since nan-ira goal is to maka-reproduce, ngani, di na hira iti-take over na nira ang cell. Then, di na hira makakag-cause han diseases, ha plants, ha animals, ngan human. When we say viroid, it is similar to virus, pero much smaller a virus than Yrikka capsid. Viroid is discovered by the scientist in 1970. Viroid can affect crops like tomatoes, potatoes, and fruit trees. So the viroid waray niya epekto ha human, pero dakot iya epekto ha crops. Pwede mag-result han crop failure ha agriculture industry. Viroid is spread through pollens, and seeds. Then, nakakag-spread gyapya pag ang farmer naggamit yahan tool, farming tool, usa nga plant nga infected na may na viroid. So, pag ginamit niya, pag ginagamit niya utro nga ito nga tool, pakadtuha usalat nga plant na waray pa viroid, so maapektuhan. That's how the viroid nakakaapekto iba. Prion, it's doesn't have neither DNA or RNA, but it is an abnormal mutation of a usually harmless protein. So, pag ang protein naging prion, it can cause a degenerative brain disease, or hayop is known as the mad cow disease, pero tao is Creutzfeldt-Jakob disease. Once the prion enters the brain, they force the normal cellular protein to begin folding into abnormal shapes. This damage the neurons and eventually leads the brain riddled with holes. The prions what niya treatment. Prions are resistant to heat, radiation, and chemicals. Cellular infectious agents, they are those microorganisms that namimilingiran host cell nga ira i-invade ngan hiran at ma-take over nga niamot mabulig ka ira nga mag-reproduce. So, may the virus, may viroids ngan prions. Although pares hira cellular, but ibay-bahira kung ngayon hira na infect and virus sa tao, usually good tao, pwede la tayo. Then viroids is only in plants. Then prions and animals. Although may the cases na may the prions nakaka-invade ka ng tao, but rare. We all know that there are 3 domains of life: the DomainBacteria, DomainEukaryote, and the DomainArchaea. Now we're going to talk about the DomainBacteria. When we say bacteria, they are unicellular organisms, and their cells are described as prokaryotes because they lack a nucleus. Most bacteria have peptidoglycan cell wall, and they divide by binary fission, and they possess flagella for motility. According to the way their cell structure stains, bacteria can be classified by gram-positive or by gram-negative by using the gram staining. DomainBacteria includes prokaryotes that people encounter on everyday basis. Most bacterial species are heterotrophic, that is, they feed on organic matter. The largest number of bacteria are saprobic, meaning that they feed on dead or decaying organic matter. A few bacterial species are parasitic. This means that they live within the host organisms and cause disease. Certain bacteria are autotrophic, which means that they synthesize their own food. Such bacteria engage in the process of photosynthesis. Bacteria may live at a variety of temperatures. Bacteria who live at a very cold temperature are psychrophilic. Those who live in human body temperature are mesophilic, and those who live at a very high temperature are thermophilic. Bacteria that requires oxygen for their metabolism is referred as aerobic, while some bacteria that thrive in an oxygen-free environment are said to be anaerobic. Some bacteria that can live with or without oxygen, they are described as facultative aerobic. Most bacterial species live in a neutral pH environment. By the way, the neutral pH is 7. And some can live in an acidic environment such as yogurts and sour cream. And some species live in alkaline environments. Certain bacteria are known to live at the pH of 2 found in the human stomach. Bacteria play many beneficial roles in the environment, such as they are the one responsible for the decay of the landfills and the other debris in the environment. This bacteria recycles the essential elements in the organic matter. In food industry, the bacteria are used to prepare foods such as cheese, fermented dairy products, and pickles. In other industries, the bacteria are used to produce chemicals, dyes, numerous vitamins and enzymes, and a number of insecticides. Today, they are used in genetic engineering to synthesize certain pharmaceutical products that cannot be produced otherwise. Bacteria reproduce by binary fission. In this process, the bacterium, which is a single cell, divides into 2 identical daughter cells. I've already discussed about the domain bacteria, so now I'm going to discuss to you about the DomainArchaea. DomainArchaea is relatively new classification of life, which is initially proposed by Carl Woese, an American microbiologist, in 1977. He found out that bacteria, which are prokaryotic cells without nucleus, can be divided into 2 distinct groups based on their genetic material. Both bacteria and archaea are single-cell organisms, but archaea have a completely different cell membrane structure that make the archaea to live in extreme environment. On the tree of life, cells of the DomainArchaea are situated between cells of bacteria and those of eukaryota, which includes multicellular organisms and higher animals. Archaea reproduce asexually through binary fission, which means that the cell is split into 2 like of the bacteria. In terms of their cell membrane and chemical structure, archaea shares features with the prokaryotic cells. The structure of archaea are prokaryotes, which means that the cell don't have a nucleus or other membrane-bound organelles in their cell. Like bacteria, archaea have the cell of the archaea have a coiled ring of DNA, and the cytoplasm contains ribosome for the reproduction of cell protein and the other substances the cell needs. Unlike bacteria, the cell wall and membrane of archaea can be stiff and give the cell a specific shape such as flat, head-shaped, and cubic. Archaea species share common characteristics such as shape and metabolism, and they can reproduce asexually through binary fission just like the bacteria. The DNA of the archaea are simpler than that of eukaryotes and similar to the bacterial gene structure. Where do archaea survive? The main differentiating characteristic of an archaea is their ability to survive in toxic environments and extreme habitats. Depending on their surroundings, the archaea are adapted with regard to their cell wall, cell membrane, and metabolism. Archaea can use a variety of energy sources including sunlight, alcohol, acetic acid, ammonia, sulfur, and carbon fixation from carbon dioxide in the atmosphere. The archaea cells are able to live in extreme environments can be classified depending on their ability to live in a specific condition. Such four classifications are tolerance for high temperature, hyperthermophilic, able to live in acidic environments, acidophilic, can survive in highly alkaline liquids, alkaliphilic, tolerance of high salt content, halophilic. The archaea possesses the following characteristics. First, the archaea are prokaryotic cell. Second, unlike bacteria and eukarya, archaea have membranes composed of hydrocarbon chains attached to glycerol by either linkages. Third, the cell wall of archaea contains no peptidoglycan. Fourth, archaea are not sensitive to some antibiotics that affect bacteria, but are sensitive to some antibiotics that affect eukarya. Fifth, archaea have rRNA that is unique to the archaea as indicated by the presence of molecular regions distinctly different from the rRNA of bacteria and eukarya. Now, I'm gonna discuss about diversity of microorganisms. When we say microorganisms, they are those organisms that cannot be seen by the naked eye, but needed the use of a microscope. So, let's tackle about algae, protozoa, fungi, lichens, and slime molds. So, what exactly are algae? When we say algae, they are those photosynthetic and eukaryotic organisms. The study of algae is called phycology, and the person who studies algae is called a phycologist. So algae and algae cells. So why is that we can say na eukaryotic and algae? Because algae may niya cell wall, cell membrane, nucleus, cytoplasm, ribosomes, plastids, mitochondria, and Golgi bodies. In addition, some algae cells have pellicles, stigma, which is a light-sensing organelle, and flagella. Although algae are not plants, but they are more plant-like than protozoa. But we can differentiate algae from plants because algae lacks true roots, stems, and leaves. Algae produce their energy by photosynthesis using energy from the sun, carbon dioxide, water, and inorganic materials from the soil to produce cellular material. Pero some species of algae use organic material, nutrients, and nakakasurvive bisan gutilaan sunlight. Most algae cells contain cellulose, polysaccharide, which an ibang microorganisms waray taira cell wall. Depending On the photosynthetic pigments they possess, an algae can be classified as green, gold, or golden brown, and red. Algae range in size from tiny, unicellular, and microscopic organism to large, multicellular, and plant-like seaweeds. Diatoms and dinoflagellates are the example of microscopic algae. Diatoms, they are tiny, usually unicellular, and lives in both freshwater and seawater. Diatoms are used in filtration aid and mild abrasive in products such as metal polishes and toothpaste. Dinoflagellates are tiny, unicellular, flagellated, often photosynthetic algae. So dinoflagellates, they are responsible for what we know as red tide. Algae are very easy to find. They include large seaweeds on various colors, brown kelps along the ocean shore, the green scum floating on the pond, and the green slippery material on wet rocks. Algae are important source of food, iodine, and other minerals, emulsifiers for pudding, and stabilizers for ice cream and salad dressing. It is also used as a gelling agent for jams and nutrient media for bacterial growth. On the downside, damage water system is frequently caused by algae clogging filters and pipe if many nutrients are present. One genus of algae, Prothotheca, is a very rare cause of human infectious disease, causing disease known as prothothecosis. The Prothotheca lives in soil and can enter in wounds, especially those located on feet. If the organism enters the lymphatic system, it can cause a debilitating, sometimes fatal infection, especially for those immunosuppressed individuals. Algae in several genera secrete substance, which is the phototoxins that are poisonous to humans, fish, and other animals.
We're done already talking about algae, so now let's proceed to protozoa. Protozoa are eukaryotic organisms that together with algae are classified in the second kingdom, Protista, of the five kingdom system of classification. The study of protozoa is called protozoology, and the person who studies protozoa is called protozoologist. Most of them are free-living organisms, found in soil and water. Protozoal cells are more animal-like than plant-like. All protozoal cells possess a variety of eukaryotic structures and organelles, including cell membranes, nuclei, endoplasmic reticulum, mitochondria, Golgi bodies, lysosomes, centrioles, and food vacuoles. In addition, some protozoa possess pellicules, cytostomes, contractile vacuoles, pseudopodia, cilia, and flagella. Protozoa have no chlorophyll and therefore cannot make their own food by photosynthesis. Some ingest whole algae, yeast, bacteria, and smaller protozoans as source of nutrients, whereas others live and dead, decaying other organic matter. A typical protozoan life cycle consists of two stages, the trophozoite stage and cyst stage. The trophozoite is the motile, feeding, divine stages in the protozoan life cycle, whereas the cyst is a non-motile, dormant survival stage. In some ways, example, the presence of thick outer wall, cysts are like bacterial spores. Some protozoa are parasites. Parasitic protozoa break down and absorb nutrients from the body of the host in which they live. Many parasitic protozoa are pathogens, such as those that cause malaria, giardiasis, African sleeping sickness, and amoebic dysentery. Other protozoa coexist with the host animal in a type of mutualistic symbiotic relationship, a relationship in which both organisms benefit. A typical example of this, a symbiotic relationship, is a termite and its intestinal protozoa. The protozoa digests the wood eaten by the termite, enabling both organisms to absorb nutrients necessary for life. Without the intestinal protozoa, the termite would be unable to digest the wood that it eats and would starve to death. Protozoa are sometimes classified taxonomically by their mode of locomotion. Some move by pseudopodia, others by flagella, others by cilia, and some are non-motile. Amoeba or amoebas such as acanthamoeba, etamoeba, and negleria move by means of cytoplasmic extensions called pseudopodia or false feet. Ciliates such as Balantidium and Paramecium move about by means of large numbers or hair-like cilia on their surfaces. Flagellated protozoa or flagellates such as Trypanosoma, Trichomonas, and Giardia move by means of whip-like flagella. Babesia, Cryptosporidium, Cyclospora, Plasmodium, and Toxoplasma are examples of sporozoan protozoa that cause human infections. Sporozoan protozoa are non-motile. Let's talk about fungi. So, what is fungi? Fungi are a diverse group that are now classified across 3 kingdoms. Those that are pathogenic to humans and animals are placed in the kingdoms of fungi. This is also called Eumycota. The study of fungi is called mycology, and the person who studies fungi is called mycologist. Fungi are found almost everywhere in Earth. Some, the saprophytic fungi, are living on organic matter in water and soil, and others, the parasitic fungi, living on and within animal and plants. Some are harmful, whereas others are beneficial. Fungi also live on many unlikely materials, causing deterioration of leather and plastics and spoilage of jam, pickles, and many other foods. Beneficial fungi are important in the production of cheeses, beer, wine, and others food, as well as certain drugs. Example is immunosuppressant drug cyclosporine and antibiotics penicillin. It is estimated that fungi are the most diverse group of organisms on Earth and include yeast, molds, even mushrooms. As saprophytes, the main source of food is dead and decaying organic matter. Fungi are garbage disposers of nature and vultures of the microbial world. By secreting digestive enzymes into dead plant and animal matter, they decompose this material into absorbable nutrients for themselves and other living organisms. That they are the original recyclers. Imagine living in a world without saprophytes, stumbling through endless piles of dead plants and animals and animal waste products. Not a pleasant thought. Fungi are sometimes incorrectly referred to as plants, but they are not. On one way that fungi differ from plants and algae is that they are not photosynthetic. They have no chlorophyll and other photosynthetic pigments. The cell walls and algal and plants contain cellulose or the polysaccharide, but fungal cell walls do not. Fungal cell walls do not contain a polysaccharide called chitin, which is not found in the cell walls of any other microorganism. Yeast and Microsporidia are unicellular, whereas molds are multicellular. One of the ways in which fungi reproduce is spores production. The 2 general types of fungal spores are sexual and asexual spores. Asexual fungal spores are known as conidia, and spores depending upon how they are formed. Yeast are microscopic, single-celled organisms that usually reproduce by budding. C. albicans and C. neoformans are examples of yeast that cause human infections. Many of the commonly used antibiotics are produced by molds. Dimorphic fungi can live either as yeast or as molds, depending on growth conditions. Mushrooms, toadstools, puffballs, and bracket fungi are examples of fungi that are not microorganisms. The molds that causing tinea infections are collectively referred to as dermatophytes. Subcutaneous and systemic mycosis are more severe types of mycosis. In the mycology laboratory, yeast are often identified to the species level by determining which substrates are able to use as nutrients. In the mycology laboratory, molds are identified by a combination of microscopic and microscopic observation and the speed which they grow. So now let's proceed to lichens. Everyone has seen lichens. Usually, while hiking in the woods, they appear as colored, often circular patches on tree trunks and rocks. For many years, it was thought that lichens represent a combination of two organisms, an alga, a cyanobacterium, and a filamentous fungus living together in such a close relationship that they appear to be one organism. Recent evidence suggests that a third party, a yeast, may also be present, embedded in the cortex of the lichens. Close relationships of this type are referred to as symbiotic relationship, and the partners in the relationship are referred to as symbionts. Lichens represent a particular type of symbiotic relationship known as mutualism, a relationship in which all parties benefit. Lichens are classified as protists. They are not associated with human disease, but substances produced by lichens have been shown to have antibacterial properties. So, always remember that a lichens is a combination of two or three organisms, an alga or a cyanobacterium, a fungus, and a yeast. So, the last topic of diverse microorganism is the slime molds. So, slime molds, which are found in soil and on rotten logs, have both fungal and protozoal characteristics and have recently been transferred out of kingdom fungi and placed in the kingdom protozoa. Thus, they are not molds. Slime molds have a very complex life cycle, initially starting as an amoeba but progressing into a multicellular organism. They are not known as cause human disease. So, thank you for listening. Hope you learned something about diversity of microorganisms.